There we go.
Wonderful. Thank you. So my name's Savannah. I am a UHN alumni. I did part of my PhD and postdoc in Toronto. And during that time, I had the luxury of working with some spatial omics data sets, including the Visium V2 and the Visium HD. And so today, just kind of want to give a quick overview of some of the different file outputs that you'll be receiving from your Visium HD data sets, some QC that you can perform, as well as different normalization strategies that you can consider. I'll keep this short so that we can move directly into the hands-on portion. But ideally, at the end of the lecture, we feel a bit more comfortable with the data that we'll be working with. So to begin, I'm just going to walk through some of the files and their structures within the Visium outputs. As already mentioned, there's a number of different files that you'll receive from your Visium dataset. The web summary file has already been discussed pretty thoroughly, so I won't go over it in too much detail, but essentially you're going to get some key metrics that give you a high-level overview of the quality of your data. These include just your mean reads and the total number of genes detected. There's some metrics regarding the mapping of your reads to your probes, so how well things are kind of aligned, as well as the sequencing itself. And to the right, I just want to highlight the utility of using spatial at the very beginning just to examine your QC. Shown here is the number of unique molecular identifiers, or UMIs, to your tissue section. And it can give you a sense of how clustered your cells are together or how deeply sequenced a region is. And it can also indicate if there's any aberrant debris or detection of signal outside of your tissue. When moving to the image alignment section, you can see how well your slice is captured within the fiducial frame here. You can see from the blue how much of your tissue is aligned. And it gives you some ideas of how well the tissue, your microscopy image, as well as your cytosys image are aligned. Next is the binning. So we've talked about binning a little bit, and I'll elaborate on the next slide about what binning is and what you can do with it. But this next portion just kind of examines the number of UMIs per bin as well as the mean genes. You'll note that the 8-micron bins, while having a higher proportion of tissue underneath them, do have fewer mean genes per bin. So something to consider early on. Below, I wouldn't put too much focus on the clustering that 10x will perform. I'm not really sure what pipeline they use. It could be very similar to the Seurat pipeline that we'll be using today, but in general, we can just look at our own data. It'd be more exciting. So bins, we've talked a little bit about bins. Naturally, 10x has a 2x2 micron bin, which is very tiny. However, it does provide more aggregate data forms in the 8 or 16 micron bins. And your initial inclination might be to go to the most granular bin type, so 2x2 micron. But I just want to caution that that does come with a cost. The first just being a high computational cost. The more granular you come, the bigger your data set is, and the more difficult it can be to process. And also you lose a little bit of sensitivity because you're zooming in so deeply into the tissue that you can lose some signal. Whereas if you aggregate it into an 8 or even a 16 micron bin, you actually enrich for your signal and may be able to capture lowly expressed genes and things that would otherwise be missed. With the 2x2, you of course are able to achieve more of a subcellular localization, but again, because it's in a grid format, you might be capturing two cells in one. So it doesn't guarantee that you get the subcellular localization within a single cell. So for those reasons, we'll be proceeding with the 16 micron bins, but the data set does come with all bin types, so you can definitely explore and decide for yourself what you would prefer. Next up, you have your bin outputs. So now that we've decided that we want to use these 16 micron bins, I just kind of want to highlight how the data is structured if you are to download it. Within the 16 micron bin folder, you see a number of different file types. The first two that I want to highlight are your filtered and your raw matrices. So by filtered, it's just filtered for spots that have been associated with tissue. And it's considerably smaller than the raw matrices, so I would highly recommend going with that. And if you were to just select the filtered matrices, you see zipped files for the barcodes as well as the features, so the genes and the matrix of the two, which you can import individually or as an H5 file. So this file format comes with multiple different files that can be imported directly into your RStudio. So it's a little bit faster and it's a bit more compressed, so it's a little bit easier to manage. And so we have our files, we're good to examine them. But what else is in this file folder? There's also some really important spatial features and folders and files. So shown in the pink here are just your low and high res microscopy images. This is what's going to be aligned to your cytosist image. There's also your scale factors in the JSON format. So these are important factors that better align both your spots and your pixels. Sometimes they don't perfectly align, and so it just ensures that your microscopy image aligns with what's being sequenced and can be later applied for some downstream analyses that Shamini will be going over. There's also just the fiducial frame, which helps ensure that your image is indeed captured and being sequenced correctly, as well as the alignment, again, of your microscopy and your cytosist image. So with all that, we can kind of proceed with looking at some of the QC metrics. A very common thing to examine as a form of quality control is the counts. If you want to be looking at gene expression across space, you want to see how that is influenced in your tissue. And shown here is just unfiltered counts between two different data sets. On the left is the FFPE processed nose brain section that we'll be working with. And on the right is a fixed frozen section that we'll be doing at the end of the tutorial. And while I wouldn't want to directly compare these data sets necessarily, I think it's interesting to see how different counts are presented in different tissue types and how the processing, as Melanie kind of touched on earlier, can really highly influence the quality or different aspects of your tissue. And as she also really hit home was that the way that you want to filter your data is to essentially remove human error. So there's a lot of different ways that you can kind of influence your spatial transcriptomics data. I have experienced many of these myself. And these are technical errors. So there's a lot of things that can kind of introduce variability, including a lack of exposure to the reagents. So when you're performing library prep, if certain tissue isn't exposed, there's going to be some sequencing errors, maybe a reduction in the depth. If you're working with really fragile tissues, in the past I've used a lot of organoid tissues. They're really subject to shearing, they fold, they're really challenging, and that is a very clear technical error that can be introduced. But I also want to highlight that there's a lot of biologically relevant artifacts, you could call them, that will influence your data. This includes how permeable your tissue is to the library prep solutions, as well as the localization of specific cells. So here's just some Nissl staining on the rat hippocampus. You can see the clear localization of clusters of cells. On the right is a mouse tissue where you can see within like the nuance staining that there's a high degree of motor cortical neuron or motor neurons at the bottom here, and within the dorsal horn you see a lot more of the smaller neurons. So cells are going to differ in terms of their distribution, their size, as well as their RNA content, right? So if you think of something like a B lymphocyte that's constantly producing antibodies, its ER size is going to be really big. And shown here is just a family of proteins, the septin proteins in young versus old hematopoietic stem cells. And you can appreciate that these different cell types have not only different distribution, but quantity of these different genes. So there's going to be so many variables that do influence your data that your end goal should just be to reduce the technical variation while preserving the biological heterogeneity. One thing to note is it's really important to understand your data type. So a lot of traditional single-cell RNA sequencing filtering methods include the filtering out of high metabolically active cell types, but more and more studies suggest that these might actually be biologically relevant if you're studying certain disease states. So just keep that in mind when you're assigning your different filtration methods. Also keep in mind that there's so little that's reported within the literature. This was just looking at different Visium V2 data sets, but you can tell that's just roughly half of the reported data sets here have actually discussed or reported their QC metrics. So consider that when you're trying to look to the literature as the gold standard, it's not quite there. I do think in time that there's going to be a higher precedent for transparency in reporting your QC metrics, um, but yeah, it's important to know that there's very little that's kind of out there. On the right, it's from the same paper. It's looking at the reported normalization strategies for your data set with the SCT transform uh, package from the Surratt team coming in to number one, but touch on that in a second. I don't think I need to convince you that normalization is important for data analysis, but I do kind of want to highlight that the end goal is just to mitigate that technical variation, right? So anything that from the user error standpoint might mitigate your tissue having equal exposure to your reverse transcription reagents or your sequencing reagents, what have you. So the end goal is to be able to compare to populations or samples through normalization. And the original form of normalization, or a very popular method that was brought in during the bulk RNA sequencing era, is library size normalization. So the assumption here is that while you may have slight fluctuations in the expression of like individual genes, these are your biologically relevant DEGs, the total gene count should be similar between cells or samples. And so if you want to employ this, you essentially divide your raw counts by the total counts per cell or per spots if you're using Visium HD data. And then you apply the scalar factor to transform the data from raw to normalized. And if you want to reduce the skewness of the data, often a log transformation is applied as well. And this is very computationally conservative. It's easier to do, and it's what we'll be doing in the tutorial following this. But I do want to highlight some caveats to it. Mainly, the library size normalization techniques do introduce a considerable bias towards highly expressed transcripts. And if you think about it, it's in part because a highly expressed transcript is just going to drive up the value of this denominator. And if you're looking at a lowly expressed gene or a rare transcript or something like that, it's just going to dilute that out. And the resulting normalized counts are going to be, you're going to retain the variance between the highly expressed genes. But again, those lowly expressed genes are just going to kind of fizzle out and be quite low. And this is exacerbated in spatial and single cell RNA sequencing data sets where you have a high degree of dispersion. So you have a lot of zeros or low values because you are sequencing at that single cell level. So it can be exacerbated by that as well. So some kind of alternatives that are coming in are binomial regression normalization strategies. You've probably heard of the Satya Labs SC Transform. This is a very popular alternative to the library size normalization strategies because it does not assume that each spot should have the same count. And in doing so, it's able to kind of mitigate out the technical noise while still preserving the biologically relevant heterogeneity. And you can kind of appreciate that from this UMAP plot, wherein you get a tighter retention of your cell types. So their biologically relevant heterogeneity is preserved. You're able to better delineate between different cell types. And while this strategy is becoming very popular and is broadly used within single cell RNA sequencing, it also comes with some caveats. Primarily, it's very computationally demanding. It's very difficult to perform SC transform if you have multiple samples and want to look at a merged object within your analysis. And in addition, it's not currently compatible with a lot of sketch assays. So in module 3, we'll be using a sketched approach where we'll be using a training data set and projecting that data and that information back onto your main data set. Currently, that's not really supported by SC Transform. So depending on what your downstream analyses are going to be, you have to consider that as well. And More recently, people have considered the spatial component when performing normalization. So you can apply things called spatial smoothing that applies something called a smoothing kernel, just based on the neighborhood expression to kind of smooth out or sometimes apply even a gradient of gene expression. There's many different ways that you can do this. There's the Gaussian kernel smoothing, which looks at the higher influence of closer spots. It's a weighted average. There's also your k-nearest neighbors. This looks at the value of the nearest neighbors and is a little bit more uniform. I don't know if I would recommend this for neural tissue, just given the fact that it does have very distinct localization of neurons in like the cortical layers, for instance. But again, it's worth trying. And there's also smoothing approaches where you're looking at the proximity and the morphology of cells. That takes on the assumption that cells with a similar morphology should have a similar total gene count. And just below is highlighting the expression of spatially localized genes DOCK2G. This is, I think, in the optic nerve, the optic cup of mouse tissue. And you can see when you apply just the traditional normalization strategies, it seems that that signal is kind of quite dim, and it becomes enriched as you apply the more spatially reconstructed and smooth normalization strategies. And I think this will be where people move towards, just given that more and more reports do claim that using or applying traditional single-cell RNA sequencing normalization strategies does it makes it more difficult to identify spatial domains. So if you think that that's really important and you're probably using spatial transcriptomics to examine like spatial components, it may be important to kind of reassess your normalization strategies. Lastly, I just want to quickly touch on integration. So if you're thinking about incorporating multiple data sets, as mentioned, it can be very challenging to prepare all of your tissue at the same time. You can only fit your one ROI within one cytosys slide. So if you think that you're going to have batch covariates, or if you predict that by sequencing, maybe a cohort of patients, there might be patient-to-patient heterogeneity, you can integrate your data using some programs, including the Satya Labs, Harmony, things like CombatSeq. And essentially what it's able to do is it's transforming your data by looking at how the global expression changes between sets, examines the covariates, you can progress them out. And in this way, it can kind of better align your two data sets. But I will say with great power comes great responsibility. Just because you can combine data sets and integrate them doesn't necessarily mean you should. If your data sets are lacking in the majority of the cell types that they're supposed to be producing, it may not be an appropriate way to analyze your data. And from a more like computational conservative manner, you can also just analyze two data sets separately and combine later. So that's just some food for thought. With that, I want to thank you for your time. This is my contact information. I will be coming in tomorrow as well to talk about some DEG analysis. And I do want to post this lovely picture of my work to highlight that I do have some different work hours. I'm on the West Coast. So if you contact me and I contact you at a weird time, that's fine.